You still had to log down to the Moodle site.
I completely, no, so I used pollen and it didn't work.
Capital letters, dude.
Well, oh my god, I swear to god if that's it. Okay, I'm gonna try now. I've been
on
trying this whole time.
letters, didn't I?
Dude, I have been very tired since then, so.
Recovering from your awesome performance on that exam, I guess.
You didn't have to do that, man. That was, okay, that was, a lot of people struggled. I studied for 6 hours for that test and I did that. I got the whole essay question wrong. I read the graphs completely wrong. All right, you can't.
6 hours and one of them was here.
What? No, was it? Wow, you didn't have to do it to me. What did I do to you?
You studied for a test in another class in my class.
Oh, that was, hey, I gotta be in that. I gotta be on that test. I'm okay with it.
All right. Get logged in here. Okay. Now's the point where we're going to start skipping around and pulling a little bit. So by next Wednesday, read chapter 8 and chapter 11. Chapter 8 introduces the second part of Poland's book, where he goes to this farm in Virginia called Polyface Farm. It takes a very different approach to agriculture than, for example, George Naylor's farm. And then chapter 11 actually explores that farm in somewhat more detail. You're in.
I know.
Okay, good. There's stuff that you might need in there. I maybe, oh, why did I say read? Helps if I can spell. So chapter 8 is called All Flesh is Grass. Chapter 11 is called Practicing Complexity. And so, Polyface Farm is run by a guy named Joel Salatin, and he practices a brand of rotational grazing that some people here in the Kansas City area practice. It's the act of moving animals very quickly from one plot of land to another plot of land and kind of rotating different animal species through the same plot of land to basically keep the grass cropped down at a certain level, but then rotating them through to allow that grass to recover. And while those animals are there, they're depositing nutrients on the soil. And so as we talk about nutrient cycling, the way George Naylor's farm accomplishes nutrient supply to the soil and the plants is by using synthetic fertilizers. The way Polyface Farm does it is by allowing animals to fertilize the farm as he grazes them through these plots of land. So in order to put this section that we're doing on nutrient cycling into this kind of broader context, I want you to have read the section that talks about George Naylor's farm, this corn and soybean operation in Iowa, and be able to compare that to Polyface Farm in Virginia, because it's just two philosophically different ways and ecologically different ways of going about farming. So then what I want you to do in preparation for Wednesday, and I'll talk about this as I finish nutrient cycling today, I've been giving you these diagrams that show you, for example, why is it not coming up? Oh, it was there on Wednesday. These types of diagrams showing nutrient cycling, this is for nitrogen. I don't know what happened to the carbon one. I'll go back and fix that, although it should be up on the one that's online. So you can draw one of these for the Naylor farm, and you can draw one for the polyphase farm. Only in both of those cases, you'll be putting in actual animals and plants instead of this general plant and animal category that we have here and here. And so what I want you to do in preparation for class on Monday is take my computer just got rid of the diagram, is to basically take these two farming systems and compare them by drawing nutrient cycling diagrams for both of those places. And you're not going to know what the amounts and stuff are because Poland doesn't tell us that, because quite frankly Poland is a journalist, he's not an ecosystem ecologist who studies biogeochemical cycles. But you can at least get an idea of how many organisms are involved in the nutrient cycling and what the kind of relative rates of flow of carbon and nitrogen are going to be through these two systems. So that's bring that into class on Wednesday in preparation for Wednesday's discussion. And what we're going to do on Wednesday is we're going to just discuss comparing and contrasting these two different ways of thinking about agriculture. On Monday, we are hopefully going to have a good, rousing discussion of genetically modified organisms as a result of having read and discussed the National Academy of Sciences report. Once again, those are short presentations where you're basically kind of boiling down your chapter for the rest of the class. So you should concentrate on what the major findings were and what the evidence for those findings was. And if you can communicate that effectively in 15 minutes, then you're good to go. You can have visual aids if you want to, but I don't know that visual aids are going to be all that helpful for you. I think what would be really helpful is having a nice, well-developed outline. If you want to bring a hard copy of the outline for the rest of your peers, that might be useful to them as they begin to prepare their written thing. By Monday, I will have figured out kind of what I want as a due date for the position paper that I want you to write on that, and I'll also give you instructions on that assignment on Monday. So Monday is going to be a good day to be here. Glad we didn't do it today. So, okay, any questions about carbon cycling? Yeah.
So for the thing prepared on Wednesday, do you want it to look like the chart up there?
Well, it can be hand drawn, but yeah, something like that. Basically, the way I divide the world is soil and things that are above soil. And so there are things that happen in nutrient cycling that go on in soil. There are things that go on in nutrient cycling that happen above the soil level. And so this whole big box here is soil. Animals are on top of it. Plants are partly on top of it, but partly down in it, whether you're talking about shoots and roots. And then you have the atmosphere out here, which is a big pool in many cycles. And so then the question is just, what are subcompartments of soil? And in the nitrogen cycle, we're going to see that there are a lot of subcompartments of soil. In the carbon cycle, there wasn't really a lot of compartmentalization within soil. Soil from a carbon perspective can just be thought of as this big thing filled with microbes and bugs that decompose things. It's a bit more complicated in nitrogen, which is why we went through carbon first. And then the question is, what are the fluxes that are going on, and which of those fluxes are transformations versus just the transfer of matter in the same form from place to place. So, and I always draw these first and foremost by hand. The ones that I have in the presentation are nice and pretty because I used a software package to build them. If you want to make them nice and pretty, there is a software package called View. I forget what university produces it. It's called the Visual Understanding Environment. It's a concept mapping program. I use it with concept mapping with my Biology 133 students, but you can also use it for building those types of diagrams that I showed you, because it allows you to put boxes in here that are topics. You can label them, but then it also allows you to make connections, and some of those connections can be arrows indicating the direction of flow of nutrients. And then there's also a spot for label in the middle of that flow where you can label what the flow is. And so they're really useful for making nutrient cycling diagrams. It's just as easy to just draw the thing on a piece of paper, which is
what I'm going to do.
What it was. Which is what I always do.
About how pretty it is.
Yeah, I did it this way so that you could be sure and read it and everything else, because I think I have reasonably decent handwriting, but in any event, you can't read it. It's it's there for you in a nice, pretty form. Other questions about carbon cycling? Okay, nitrogen is harder. I'm just going to preface what we're saying today. Nitrogen cycle is probably the most complicated biogeochemical cycle you can deal with. But it's crucially important because crops like corn use a ton of nitrogen, and crops like soybeans replenish nitrogen to soils because they are nitrogen-fixing organisms. So, we've already talked about the presence of nitrogen in the atmosphere. It makes up a huge portion of the atmosphere, but it's found in what we refer to as diatomic nitrogen, these two nitrogen atoms bonded together by this triple bond. The problem with this triple bond is that it is very, very strong, and so it takes a lot of energy to basically break that bond, combine that with hydrogen to make, in this case, ammonia. So the things that you need to think about and a way of organizing your knowledge where nitrogen cycling is concerned is to think about transformations. So this is the first transformation that we have to deal with. This is a transformation from atmospheric nitrogen to ammonia, NH3. Don't memorize the entire equation, just know that you're going from this starting form of nitrogen to this ending form of nitrogen. For completeness, that's the chemical equation, but this is essentially what you need to know. This is nitrogen fixation.
Okay, so I'm just dropping this off real quick.
Yeah, okay. So this is the first form of nitrogen down the path of making nitrogen useful to plants. Things that you need to know about nitrogen fixation is that it is accomplished by bacteria in soil, and it can be accomplished by bacteria in things called rhizobia in legumes. This is why I wanted you to read the chapter about legumes, is because legumes have these root nodules on their roots that contain nitrogen-fixing bacteria that invaded the plant roots from the soil, and these nitrogen-fixing plants, these nodules, is where a lot of biological fixation of nitrogen happens. So, it's a process by which atmospheric nitrogen is converted into ammonia. It's carried out by bacteria in association with plants and freely in the soil. These bacteria are what we call heterotrophs, meaning that they consume lots of different kinds of substances to get energy. So most of them are heterotrophic. Cyanobacteria also do some nitrogen fixation. These are autotrophic, meaning that they do photosynthesis. And so most of the ones that we're talking about are these heterotrophic soil bacteria. So, and it's made available to plants, and the plants use this nitrogen that is necessary for amino acids and nucleic acids primarily. The problem with getting atmospheric nitrogen into the form of ammonia is that this requires a lot of energy. And when humans do this, we burn a ton of fossil fuels to make nitrogen fertilizer when we do the Haber-Bosch process. The Haber-Bosch process requires that you do this under high heat and high pressure. And so, because the Haber-Bosch process is a primarily chemical process, it gets all of its energy for breaking the bonds of N2 through heat and pressure. The way organisms do this is because you can't just heat up a bacterium to really hot temperatures. We do this through enzymes, which we've talked about before. Enzymes generally lower the energy of activation that is required for a reaction to occur, and the enzyme that is used for this is called nitrogenase. Remember that all enzymes end in ASE. see something ACE, that's an enzyme. And this is the enzyme that facilitates bacteria being able to basically break this triple bond of nitrogen, allow hydrogens to then, if you have 3 bonds between nitrogen and another nitrogen, when you break nitrogen in 2, you now have 3 electrons free, and you can attach a hydrogen to each of those. Doing the attachment of hydrogen is pretty easy. Hydrogen will naturally want to join some free electrons to complete its outer shell of electrons. The trick isn't getting hydrogen to bind. The trick is freeing up the 3 electrons of nitrogen so that they're capable of binding, so that the energy is required to break the bonds of N2. Bacteria do this at much lower temperatures because they have this enzyme called nitrogenase. So these bacteria originally started out as soil bacteria, and what they do is they then invade the outer cells in the epidermis of the root of a cell, of a plant rather, and when they invade the cell, they then, essentially through manipulating the plant, they encourage that plant to essentially build up plant material around the bacterial cell, and you end up with these root nodules. I meant to do this today. I have some soybeans growing in my backyard, and I meant to bring in a soybean plant so you could actually see the root nodules, but this is what they look like. And if you cut these open, they look reddish or pinkish inside because they have a protein in there called leg hemoglobin, which also helps facilitate this process. And so you have this mutualistic interaction between these nitrogen-fixing bacteria and the plant. The plant provides a place for them to live, and the bacteria undergo nitrogen fixation. So in this relationship, why would bacteria do this for plants, and what are plants giving bacteria, and what do the plants themselves get? So whenever you have a mutualism, that's generally, mutualisms are interspecific interactions where species A, let's say it's the plant, and species B, in this case we'll say it's the nitrogen-fixing bacteria, they both get something out of the deal. There are other interspecific interactions, like predation is an interspecific interaction, and in this case, the predator gets something out of the interaction and the prey gets killed. Parasitism is another one, where the parasite gets something positive out of the interaction and the host suffers negative consequences for having a parasite. There is commensalism, where one species benefits and there's no consequence at all for the other individual. So we can classify interspecific interactions on the basis of what happens to the 2 species. In competition, for those of you in business, competition in ecology is defined as an interspecific interaction where both species incur negative impacts. Why do people like Elizabeth Warren want to increase competition, for example, for healthcare? Or increase competition for anything? Why do some people want to break up monopolies? Why do some people want to break up Google? Google has been in the process of acquiring all kinds of things. What all kinds of things does Google own?
Everything almost.
Yeah, okay, so Google has done that. I guess I can remember more of Facebook's acquisitions. So Facebook now has Instagram. Facebook has, what else does Facebook control?
Facebook has Facebook, Instagram. I know there's one more I'm thinking of. I just saw it recently.
Didn't they get MySpace too?
MySpace.
Yeah.
Who uses
Did they
MySpace?
Did they buy
They bought
MySpace?
MySpace.
They bought out a
So-
competitor. Why do companies want to buy out competitors?
So don't have to worry about losing.
So they don't have to worry about losing market share.
They can add on, they can like absorb those customers who use MySpace.
They can absorb those customers, and then there's no place for customers to go other than Facebook. You can't go to MySpace anymore because MySpace is just Facebook.
Wouldn't that be a plus? rather
That's
than-
great for, so that's actually reducing competition.
Oh, they
So-
have YouTube.
Oh, they have YouTube also.
That's what it was, YouTube.
Okay. YouTube is a Facebook thing. So now, video sharing is owned by Facebook. Photo sharing is owned by Facebook. Facebook is owned by Facebook. MySpace is owned by Facebook. Is there anything Facebook doesn't own? So, by reducing competition, what you do is you become the only game in town, and so you don't suffer the negative consequences of competition. Some people want to do things that increase competition, like doing things like breaking up Facebook so that it doesn't have a monopoly on all media, because competition then forces them to be in competitive interaction with another set of companies, causing them to have better practices in terms of privacy. They make less money, but as a result of that, probably prices are cheaper for advertising if you have multiple places to go and advertise. Facebook makes all of its money through advertising. And so if you can only advertise on Facebook or any of its subsidiaries, then they can charge whatever they want for advertising. If there's another game in town, they have to cut their prices in order to compete. So ultimately, competition is bad for corporations. Who's it good for?
Consumer.
It's good for the consumer, because consumers in a competitive market generally get lower prices for goods and services than they would in an uncompetitive environment. So competition, this is corporation A and corporation B. It's bad for them because they generally have to offer goods and services to consumers at a lower price. It's good for consumers for precisely that reason. So you can think about competition in terms of organisms out in nature. You can also think of competition in terms of businesses and corporations. So these are interspecific interactions, and we can classify them on the basis of whether there are positive or negative consequences for the two species that are involved in the interaction. We're talking about the mutualism here, where we have a mutualism between plants and bacteria. What's in it for the two species?
Plants get the nitrogen.
Plants get the nitrogen, that's easy. The bacteria being in association with the roots, when they fix the nitrogen, the plants can immediately suck up those nutrients and put them into their bodies, building whatever it is that they need to build that requires nitrogen. What do the bacteria get out of the deal?
A place to feed.
Okay, what are they feeding on? Where are bacteria getting their energy? Where does everything get energy? What's the molecule that stores energy in organisms?
Fat.
6 carbons, comes from photosynthesis.
Mesophyll?
Nope, those are cells.
So glucose.
Glucose.
That's what
Who makes glucose?
Plants.
Plants. So who has the surplus of glucose that gets sent down to roots for storage? Plants do. Guess where the rhizobia are? These nitrogen-fixing bacteria are.
In the soil.
They're not in the soil. They are. They're in the roots. And so what are bacteria getting out of the deal?
Glucose.
They're getting glucose. They're getting energy. So the bacteria are getting what they need. They need energy to survive. The plants are then getting what they need, which is the nitrogen that they need to survive. Rhizobia couldn't, the nitrogen-fixing bacteria couldn't survive without energy. The plants could survive, but not as well without the nitrogen. So each of them gets something that they need, and they have to give something up in the process. The bacteria give up nitrogen, the plants give up glucose, the bacteria gain glucose, and the plants gain nitrogen. This is the very nature of mutualism. If only Congress worked so well, or any form of government in the United States right now. So this mutualism allows each of these two species to thrive, and it really benefits the nitrogen-fixing plants. Because these plants, legumes primarily, fix nitrogen, this is one of the plants that farmers use when they do crop rotation, where they'll grow one crop that consumes nitrogen for a few years, and then they'll rotate that out and rotate in a nitrogen-fixing plant that also is a crop species as well. And so throughout the Midwest, you see a lot of corn and soybeans being grown, and what you'll see is that if you bike by the same field year after year like I do, so I have a set number of bicycle routes, and they go around small roads here around the Liberty area. And so I see what happens with farmers' fields over time, and you'll see a farmer plant corn for 2 or 3 years in a plot of land, and then the next year you'll see them plant a crop of soybeans, and then they'll harvest those soybeans. They till those soybeans under the following spring, and all the nitrogen that is trapped in the roots and the bodies of those plants then gets returned to the soil through decomposition. And then after that soybean crop, they'll plant corn for another 2 or 3 years, and then they'll rotate in soybeans again. And so this is a way in which you can increase nitrogen content to the soil. Nitrogen fixation, taking nitrogen out of the air, getting it into the form of ammonia. The trick, though, is that plants don't readily absorb ammonia. Plants readily absorb ammonium and nitrate. So this nitrogen fixation, this initial fixation of nitrogen, is just the beginning of the process because it's still not in a form that is super readily available to plants. So plants benefit from getting nitrogen. The bacteria benefit from getting energy. We do a lot of industrial fixation. At this point, we actually do as much fixation as a species doing industrial fixation as all of the natural fixation that goes on in the world. And so we are major disruptors of nitrogen fixing because of cultivation of nitrogen fixing crops, industrial fixation by the Haber-Bosch process, and also when we burn fossil fuels, we create nitrogen oxides that go into the atmosphere that fall back down into the earth in the form of rain, and then once they're in the soil, then the normal processes by which soil bacteria process nitrogen can take place. And so that nitrogen can also become available to plants as well. And so a lot of this comes in the form of rainfall and dryfall, dust and things like this falling down onto the ground. So we have disrupted the nitrogen cycle quite a lot. There are nitrogen transformations that aren't really changing the form of nitrogen. So if you have a plant growing on the surface of the ground, it dies, it falls to the ground, and it goes through the process of decomposition. That first decomposition is just going to basically be breaking down the major molecules into subsequent smaller molecules. And so if you have a lot of nitrate in that plant, the stuff that comes out of that is going to ultimately be nitrate. And so this act of going from living organic matter to dead organic matter is death and litter-fall, and then decomposition is the process of going from dead organic matter to dissolved organic matter, dissolved in water. But once it's in the dissolved form, it can be subject to all sorts of things that move the nitrogen around. Leaching, we've talked about. Leaching is done by what molecule? It's a polar molecule, it's everywhere. It's done by water. Why does water do leaching?
Because it drags nutrients with it.
Yeah, it's partially positive and partially negative. Partially positive on the hydrogen, partially negative on the oxygens. and so it can grab onto other charged molecules. NH4 is a positively charged molecule, NO3 is a negatively charged molecule, and so both of these things can be leached out because water will tend to bind to them and drag them through the soil. So leaching, fragmentation of dead organic matter, changes in chemical and physical structure. A lot of organisms will ingest plants, and in the process of ingesting them and digesting them, they'll actually add microbes to the plant material that they're eating, and the stuff that they poop out actually decomposes faster than the stuff that they would have brought in. So we have bacteria, fungi, and all sorts of little small insects and other arthropods. And so this process of decomposition occurs on the surface of the soil and deeper in the soil, and it's influenced by temperature, the amount of moisture that is there. You can have too much moisture or too little. If you have very little moisture, decomposition slows way down. If you have too much moisture, decomposition slows way down because you don't have enough oxygen in the soil for decomposition to occur. Because remember, decomposition is just taking things like glucose, adding oxygen to it to break it down to CO2 and H2O, and releasing heat. And so this is how decomposition works on carbon-containing molecules, and you need oxygen present. If you have a waterlogged soil, there's no oxygen or very little oxygen in the soil, and so decomposition slows down. And carbon is actually what gets in the way of decomposers getting to nitrogen. So we have this dissolved organic nitrogen dissolved in water in soils, and that gets combined with hydrogen ions that come from the moisture in the soil. So you can think of water as this, but you can also think of water as dissociating. Remember that these represent 2 electrons. One electron that came from oxygen and 1 electron that came from hydrogen. What does oxygen tend to do to the electrons of hydrogen? Pulls them away. And if it pulls it away enough, you can get a hydroxyl ion with a full negative charge, leaving this hydrogen without its electron, giving it a full positive charge. These positive charges can then combine with NH3 to create NH4, which now has a positive charge because it picked up this positively charged ion that came from water. This is ammonium, and this process is called ammonification. So in the same way that we did this for nitrogen fixation, we have NH3, which came from there, and this process is called ammonification. Why is it called ammonification? Because it produces ammonium ion, which is a positively charged ion. Now this can be absorbed by plants. Yes?
Okay, so ammonium.
Mm-hmm.
The Haber-Bosch process creates ammonium, right?
It creates ammonium nitrate.
Ammonium nitrate, and that's what we like weaponized for World War II.
Yeah, so that's a major constituent of gunpowder.
Right, but ammonium is like a natural thing too. It's
Oh,
not
yeah.
something we like. Okay.
So we can create it in a lab through
Right.
chemical processes in a lab using something like the Haber-Bosch process, or it also occurs naturally. You buy it when you go to the store and
It's like
get
in cleaning
cleansers. Yep, it's in
products.
cleaning products.
Okay.
Yeah. So ammonium is a weak acid, and so that's why we use it in cleaning products oftentimes. And so, yeah, it has lots of uses. It's not just used to blow things up.
Okay. So we definitely like still use the Haber-Bosch process to make the
So
ammonium nitrate.
when you have ammonium and you have nitrate, because this is a positively charged ion and this is a negatively charged ion, they will naturally come together to form ammonium nitrate. The big trick with the Haber-Bosch process, though, was cracking this first nut,
Okay.
breaking the bonds of nitrogen to produce ammonia. Once you have ammonia, It's dirt easy to get ammonium from ammonia, and it's dirt easy to add that to nitrate. The tricky part was this part.
Okay. But the plants were already making like the ammonium nitrate.
Plants already do this.
Yeah.
Well, bacteria
Bacteria, yeah.
associated with plants already do this.
Okay.
Yeah, don't confuse that the plant is doing this. The plant is providing a home for bacteria, and it's actually the bacteria that
Okay.
do the work.
Okay.
That's something I'll even slip into talking about nitrogen fixing plants. Legumes aren't nitrogen fixing plants. Legumes are organisms that have nitrogen fixing bacteria in their root nodules, and as a result, they get the benefits of nitrogen fixation. If I slip into saying nitrogen fixing plants, check me, because I'm meaning to say something else, but sometimes it comes out that way
Right, yeah.
because we think of them this way. So the reason I'm laying this out to you this way is this is atmospheric nitrogen that gets converted into ammonia, and then ammonia is the next step for the next reaction that gets ammonium. Ammonium can be absorbed directly by plants, but ammonium can also be combined with nitrate. So, whoa, go back. So we basically have dissolved organic nitrogen. Ammonia combined with hydrogen gives us ammonium. This is easily dissolvable in water, but it's controlled by the availability of raw material, the dead plants and animals that have nitrogen-containing compounds in their bodies. It's limited by the ratio of carbon to nitrogen. Most of plants are made up of carbon in the form of cellulose. Remember that cellulose is just a bunch of glucose molecules bonded together. Organisms have to break down that carbon-based structure to get at the nitrogen and phosphorus containing compounds that are inside those cells. Remember that the cell is surrounded by a cell membrane. This cell membrane is made up of phospholipids. So there's a phosphorus molecule as well as a lipid molecule in this. There are proteins floating around in this cytoplasm. There's a nucleus in here that has nucleic acids, which are also nitrogen containing. But around all of this is a thick cell wall. And this thick cell wall is made up of cellulose. So if you're an organism and you're trying to access the things that are inside a plant cell, you have to get through the cell wall first. And so you have to digest away all that carbon. So the more carbon you have relative to the amount of nitrogen that you have, the less interested somebody is in eating that, in consuming that. And plants do vary quite a lot in the ratio of carbon to nitrogen in their bodies. That affects the rate of decomposition because it's just harder to get the nutrients out of high carbon-nitrogen ratio plants, whereas if you have less carbon protecting that nitrogen, it's easier. And you'll actually see organisms preferentially consuming low carbon nitrogen food. Think of it this way. What would you rather eat if your goal was just to get the eating process done and over with? Would you rather eat a single lobster tail or 50 crayfish? They're both decapods. They both have pinchers. They both have a big tail. The tail is where most of the meat is. What's the difference between having a lobster tail and having crayfish?
It's bigger. Lobster tail's
It's bigger. Open up the lobster tail once and you have a nice big fat lobster tail to eat on. What do you have to do to get the same amount of energy and nutrients out of crayfish?
Eat 50
a lot
of them.
more.
You have to eat a lot of them. So if your goal is to get energy and nutrients, you're way better off going for the thing that just has less investment in terms of getting at the thing that you need. Pop open the lobster tail, you have all the nutrients and energy you need, rather than busting open 50 crayfish. Same thing with carbon-nitrogen ratios. A lot of carbon means that you, the decomposer, are having to work through that carbon in order to get to the things that you really want, which is things like nitrogen and phosphorus and the nutrients that you need, as opposed to a low carbon nitrogen ratio food that you have to work through less carbon to get to that nitrogen, the phosphorus, and everything. And so carbon nitrogen ratio can actually affect what's going on. Temperature and moisture, once again, affects all of these processes. So now we have ammonium, and the next process that we need to think about is nitrification, which is the process of taking nitrogen and through a bunch of steps getting it to nitrate. Because it adds oxygen to ammonium. This is an oxidation reaction, but we'll just call it nitrification. Nitrification, once again, is done by bacteria. It's a different set of bacteria than the bacteria that we've talked about before. These are nitrifying bacteria, bacteria that actually facilitate this chemical equation going to completion. And these bacteria get carbon from carbon dioxide and energy from this oxidation process. So they're actually getting energy as a process of doing this, and so this is just a byproduct of their own metabolism. They don't have a goal of making nitrate, it's just the process by which they go about doing their metabolism, and this is a byproduct of metabolism, just like carbon dioxide is a product of our metabolism. So Nitrous amonis and Nitrobacter, these are both nitrifying bacteria. Requires oxygen, obviously, because oxygen is in the equation, and it's ultimately limited by the starting reactants. And so from atmospheric nitrogen, you generate ammonia. From ammonia, you generate ammonium, and from ammonium, you generate nitrate. And nitrate is the most easily used form of nitrogen for plants. These are transformations that go on, and they go on primarily either in the roots of plants in the soil or in bacteria in the soil. This goes on without the need of any bacteria because that's just a natural sort of reaction of water interacting with ammonia to produce ammonium. And then this, once again, is done by bacteria primarily in soils. So a lot of the nitrogen cycle is in that soil compartment that we talk about. As a result, plants get access to that soil with their roots. And so one of the reasons that plants grow roots is to hold them in place. Another reason that plants grow roots is to absorb water. But another reason that plants have roots is because that's how they get their nutrients. They don't absorb nutrients from the atmosphere through their leaves and stems. They absorb nutrients from the soil through their roots. When you fertilize a plant, you don't spray the fertilizer on the plant. You put the fertilizer on the ground. It goes into soil solution and then can be picked up from soil solution by the roots of the plant. So plants themselves have different root structures. These are just drawings of the different kinds of root structures from different kinds of prairie plants. But there are some prairie plants that also have a type of symbiotic relationship with other organisms that interact with plant roots that help plants get nutrients. Did you guys read about that in the legume chapter? What are those plants, or what are those organisms? Across Mount Horizon. This literally means These are fungi, once again soil fungi, that basically grow in close association with plant roots. And the cool thing about fungi is normally, what's a fungus that you're familiar with?
Mushrooms.
Mushrooms. So when you go to the store and you buy a mushroom because you're making a pepperoni pizza and you want mushrooms on it, this is actually the fruiting body of the mushroom. This is the reproductive part of a mushroom. But underground, this mushroom has sent out, well actually, what has happened, the body of the fungus is made up of these long filaments that run through the soil. These filaments are called hyphae. And that's the body of the fungus. And they produce this fruiting body that we could occasionally come by and lop off and eat. And sometimes lop off and eat and die because some fungi, some mushrooms are poisonous. But this is just the reproductive structure of this organism that lives below ground. Do you guys know what the largest organism on the world, on the planet is?
Okay.
Hmm?
Is it the fungus?
It is a fungus.
Have you heard of it before?
Oh yeah, well duh,
Really?
of course I have.
Have we heard of it?
Probably not. I mean, maybe, it makes the news occasionally. Largest living organism is the Armillaria fungus. Larger than a blue whale. Covers 2,385 acres, almost 4 square miles in Oregon. It's a fungus, and it basically has sent out its fungal hyphae or fungal mycelia. These branches of the fungus that grow through the soil. And so the way they figured this out is they took soil samples from all over, and they basically sequenced the DNA that was found in the soil, and they found that there was this fungal genotype that was the same over a broad span of in this case Oregon, that is all the same DNA. And it's because in all those little soil samples there were mycelia growing through the soil, such that this one fungus, this one individual fungus, has grown all throughout the soil. And so all of the bulk of a fungus that you see is underground, and you just see the little fruiting head that pops up out of the ground when it's time for it to reproduce. And so fungi can be really big. And so these fungal mycelia are kind of the roots of the fungus, and they basically grow out into the soil, but they can also grow into the roots of plants. And when they send out their mycelia into the soil, those mycelia pick up water, and they pick up phosphorus, and they pick up nitrogen, and they will bring those things back to the plant root. You can have endomycorrhizae, which basically grow into the root, and you can have ectomycorrhizae, which basically form a crust around the outside of the root. But what these two things do, both types of mycorrhizae, essentially vastly increase the amount of soil that the plant root has access to. Plant roots send roots out, they send root hairs out, but then the fungi that are attached to those roots have much longer mycelia connections that go throughout the soil. And so you can vastly increase the amount of volume of soil that you have access to, because all of this gray stuff that's out here at the edges, this is all fungal mycelia associated with the roots, which are closer to the plant. So I've already told you the answer to this. What are the plants and the fungi getting out of this relationship?
They're getting nutrients.
Plants are getting nutrients and water.
The fungi are getting, I guess, somewhat the same.
Same as the bacteria. What are they getting?
Glucose.
They're getting energy in the form of glucose. And so in order to in order to encourage the fungus to grow in association with the plant has to give something up, and in the process of giving up that glucose, they get the benefit of nutrients being delivered to the roots by the fungi. So, as it turns out, mycorrhizal fungi are crucially important in bringing phosphorus to plant roots. And one of the things that happens when you use synthetic fertilizers on agricultural fields, if they are high in phosphorus, the plants can get enough phosphorus without mycorrhizal fungi, and they shut off the glucose spigot, essentially. They can control to some degree the degree that they let glucose loose, and what you find in farmlands that are regularly fertilized is you find the diversity of mycorrhizal fungi going down because the plants don't give up as much glucose because they can get enough phosphorus without mycorrhizae. The mycorrhizae then decline in population size because they're not getting energy from the plants because the plants are getting phosphorus from the synthetic fertilizer. And then if something happens such that you can no longer fertilize your field, your plants are now much less likely to thrive because there's not a community of mycorrhizal fungi in that field. Yeah, you have a question?
Yeah, so why don't they just like put more fungi into the like soil instead of using the phosphorus soil?
That's the trick about fungi. So these mycelia that go through the soil, they're easily broken, and once they're broken, they're no longer connected to the body of the fungus. And the other thing is they're also very difficult to culture. People who work on soil fungi really do a lot of genetic work of like taking samples of soil and then sequencing it to see what's inside of there, because it's actually very difficult to isolate individual types of fungi from soil and grow them in labs and stuff like that. So working with fungi is actually a pretty tricky thing. You might not think that because you leave some food out on your counter and in a day or two it's got fungi on it, but working with soil fungi is actually pretty challenging. One of my former students now teaches at the University of Montana, and he studies soil microbes and their role in things like decomposition and biogeochemical cycling and things like this. And he didn't start out as being much of a molecular biologist, but now almost all that he does is molecular biology and sequencing because it's just difficult to work with these organisms as organisms.
Is phosphate soil worse for the environment though, because like the runoff and stuff?
I'm sorry, what?
Is the phosphate soil worse for the environment because of the runoff?
So yeah, certainly adding phosphorus and nitrogen to soils in the form of synthetic fertilizers can be damaging to aquatic systems in particular because the water leaches through.
Yeah.
The water goes through the soil, leaches out phosphorus and nitrate. Phosphorus and nitrate are both negatively charged ions, and so water will pull those out of soil, and then that water flows into a stream. If you ever cross over a stream or a river that's kind of a pea green color, it's because chances are they've experienced runoff that's full of nitrogen and phosphorus. That feeds the algae that grow in the water, and they grow faster because they have a good source of nitrogen and phosphorus, and that's an algal bloom, which is turning the water that color. Those algae have a really fast life cycle. You can see the algae in the water column, and that's what makes it look green. But as they die, they sink to the bottom of the water, and then they go through the process of decomposition. Remember that decomposition is basically breaking down glucose, adding oxygen to it. So whenever you have decomposition happening in water, it's consuming oxygen, and then there's less oxygen available in the water for other organisms to survive, like fish and things like that. So a lot of times with nutrient pollution, you will get low dissolved oxygen in the water, and you'll get things like fish kills and things like that, simply because you just have too many nutrients in the water. Your municipal water treatment facility, when you flush the toilet, that goes to a water treatment facility, a sewage treatment facility. And that sewage treatment facility, the first thing that it does is it has big vats that basically have all of your poop and all of your pee and everything else that you flushed in it. And they have a boom that goes around, and that boom has these big metal discs with little teeth coming out of them, and they spin constantly. And they're basically spinning shit slurry up into the air to keep it aerated so that there's enough oxygen so that the bacteria that are in that vat can undergo decomposition, which requires oxygen to proceed. Once they've broken down a lot of the organic matter that is in our excrement, they move that to a series of ponds, and those ponds are designed to let algae remove the nutrients from that water, because now you've broken down all of the organic matter to all these little small organic molecules like nitrogen and phosphorus. Now you need something to absorb that nitrogen and phosphorus, so that then when you release that water to a stream, which is ultimately where it's destined to go, you're not putting a lot of nutrients into that stream. So oftentimes a wastewater treatment facility will have these big vats where they do the primary decomposition, and then they'll have a series of pools on your way to the nearest stream, and in each of those pools you'll see them. If you look at them on a aerial photograph, you'll see the ones near the slurry. Those are really green, and then they get less green and less green, and it's because as they go from pool to pool, as they move the water from pool to pool, the algae and stuff are removing more and more of the nutrients out, so then it's safe to put into the water without having a negative impact on the stream. So yeah, they can cause problems. The amount of fertilizer that we put on farms here in the Midwest that flow down the Missouri River and the Mississippi River into the Gulf of Mexico has created this big dead zone in the Gulf of Mexico that you probably have heard about in the news if you pay attention. And it's all due to this nutrient effluent coming down the Mississippi River. It leads to algal blooms in the Gulf of Mexico off the coast of Louisiana. All that stuff sinks to the bottom. When it gets to the bottom of the Gulf of Mexico, it decomposes, removes oxygen from the water. So you have this big area, and it varies from year to year in terms of how big it is. And that big area essentially is low oxygen content seawater, and as a result, stuff can't live there very well. All right, we're not quite finished, but this is basically just showing you that the root hair is the degree to which the plant has influence in getting nutrients from the soil, but then the mycorrhizal fungi can extend the reach of the roots further out into the soil, which then just gives the plant more soil to draw nutrients from. And we'll pick up on this on Wednesday. Between now and Wednesday, read the section of the chapter about nitrogen cycling from the legume chapter in Plants and Society, and look at the last few slides in here that kind of summarize the nitrogen cycle so that you can come in with some idea of how these two farm systems might differ. And we'll begin on Wednesday here. And your test is Monday after fall break. So nitrogen cycling will be the end of material from this chapter.
It'll be about like nitrogen cycling.
It'll be about nitrogen cycling, the end of genetics and genetic engineering. That'll be what's on the next test. Thank you. Good questions, by the way.